Imagine if animals could harness sunlight like plants do. Researchers have now made this a reality by enabling animal cells to perform photosynthesis. Photosynthesis in plants and algae is done by chloroplasts, tiny organelles that convert sunlight into energy. These structures likely originated from ancient photosynthetic bacteria absorbed by early cells. While animals didn't evolve with chloroplasts, scientists at the University of Tokyo have changed this by successfully implanting chloroplasts into hamster cells, which then generated energy through photosynthesis for 2 days. Professor Sachihiro Matsunaga noted this achievement as the first detection of photosynthetic activity in animal cells. Surprisingly, the chloroplasts functioned longer than expected, continuing to produce energy. Some animals, like giant clams, already harness photosynthesis through symbiotic relationships with algae. Previous attempts to create photosynthetic cells combined cyanobacteria with yeast, but animal cells posed a bigger challenge. A key issue is temperature. Most chloroplasts become inactive below 37 degrees Celsius, while animal cells require lower temperatures. Researchers used chloroplasts from Cyanidiocyzon marole, algae that thrive in volcanic springs and remain active at lower temperatures. After isolating these chloroplasts and injecting them into hamster cells, researchers found the chloroplasts were still producing energy after 2 days, even speeding up cell growth. Interestingly, chloroplasts migrated near cell nuclei, possibly interacting with mitochondria, suggesting a potential chemical exchange that requires further study. The chloroplasts started degrading after 2 days, ceasing photosynthesis by the 4th day. Genetic modifications could prevent animal cells from digesting these organelles. While the idea of sunlight-powered humans remains speculative, this breakthrough has promising applications in tissue engineering by potentially improving oxygen delivery in lab-grown tissues.